So the next one, it is in a different, completely different field. I mentioned in my opening that in order to develop drugs, if we have one target for multiple diseases, this is a very sustainable solution. Because you save the environment, you decrease the cost, and you decrease hospital time and many other. So it's a sustainable solution. So this comes from Davis Joseph with a unified theory of neurodegeneration disease rooted in axon-specific deamidation. So it is a key target to cure simultaneously Alzheimer, Parkinson, and other neurodegenerative diseases. Davis, please, on stage. Davis has 10 presentations at this summit. He has 10 papers. So this is the first one.
Can you hear? Okay, perfect.
You have forward.
Perfect. All right. Thank you very much for having me today. I will go through everything that has been discovered in my last two papers. The first paper was published in November 15, 2024. The fundamental neurobiological mechanism of oxidative stress related for EBB2 protein deamidation. The second paper being the unified theory of neurodegeneration pathogenesis based on axon deamidation, a review paper of 224 references. First off, I'd like to thank my mother. She passed away last year at the age of 50 due to breast cancer. I wouldn't be who I am today if it wasn't for her. I thank her very much for everything. So we will go through the background, the research focus, my discovery, the experiments I conducted, the experiments I invented, the theory behind my work, the links with neurodegeneration, and the next steps. So since this is a vast audience, I will briefly go through the fundamentals of biology for everyone to have a better understanding of my work. DNA, as described by the great Dr. Jehanover, is the fundamental code of life. The information in DNA is carried by mRNA, messenger RNA. Messenger RNA contains a 5' cap. The 5' cap, among other things, protects it from degradation so that the information can be carried properly. And the 5' cap during during protein production is bound to an interesting protein called the eukaryotic initiation factor 4E, discovered by Dr. Aaron Shatkin and his student, Dr. Witold Filipovic, in 1976 in their paper in PNAS. The ribosomes are then recruited by EIF4E and the EIF4F complex to produce the proteins. The ribosomes are molecular protein machines which produce everything regarding proteins, peptides, enzymes, and these little things, they make all the activity in your body. Respiration, hormones, antibodies, etc. Those are the proteins. They're involved in everything. And this leads us to the proteins I currently study at Gifu University, 4EBPs, which stop protein production by inhibiting EIF4E. So 4-EBPs were discovered by Tay and Lin in the John Lawrence lab in 1994. 4-EBPs bind to EIF4E. This binding, a process known as inhibition, stops the recruitment of ribosomes by EIF4E. As a result, proteins can no longer be produced. So you have two competing factors. EIF4E initiates protein synthesis, 4-EBPs stop protein synthesis. 4-EBP2, my protein of interest, is a member of the 4-EBP family found mainly in the brain. 4-EBP2 controls all brain activity because it's responsible for regulating the production of proteins in the brain. All chemical reactions in the brain are regulated in one way or another by 4-EBP2 in the processes of neurochemistry. 4-EBP2 deamidation is a post-translational modification, which is found everywhere in 4-EBP2 in the brain. 4-EBP2 is found in all cells of mammals. However, 4-EBP2 undergoes a chemical reaction, deamidation, as previously said, which was discovered by Birinosti et al. In 2007. This chemical reaction only happens in neurons, in no other cells. And it makes possible protein production. When you have deamidation in 4-EBP2, the 4-EBP2 protein can no longer function. It becomes obsolete, and EIF4E can function properly in the protein process, in the protein production process. 4-EBP2 is not destroyed or destabilized by the chemical reaction, but it can no longer complete its functions. Which leads to my research focus. Why does 4-EBP2 deamidation happen only in neurons? You have, like I said, 4-EBP2 is in the kidney, 4-EBP2 is in the liver, 4-EBP2 is in the pancreas, but this chemical reaction happens only in brain cells, neurons. So for 16 years, despite extensive research by the experts in the field, there was no credible explanation as to why this reaction happens specifically in neurons and nowhere else in mammals. This is very important because 4-EBP2 deamidation significantly alters protein production in the nervous system. We have to know where this reaction comes from. Any imbalance in protein production in the nervous system causes neurodegeneration. Discovering what causes 4-EBP2 deamidation would allow us to control this balance of production and have a better understanding of how brain cells are maintained and how they can continue living. This led to my work. So I came up with a credible hypothesis of why 4-EBP2 deamidation happens specifically in the brain. I thought it was because of what makes neurons unique. These cable-like structures that only brain cells have, neurons, called axons. So in order to analyze the axons, I used some experiments which are common in my field called immunoblotting, also known as Western blots. Immunoblotting separates proteins based on their weight. The heavier they are, the slower they migrate. Western blots can detect 4-EBP2 specifically thanks to antibodies which target the protein. 4-EBP2 extracted from the nervous system appears as 3 bands, as you can see here. This is a blot I did. The bottom band, no deamidation. Middle band, partial deamidation. Upper band, complete deamidation. And at the bottom we have a loading control. This is a method of analysis I invented called the deamidation ratio, where you divide the complete band intensity by the no deamidation band intensity to measure how much deamidation has occurred in the cell. I used the 2-month-old male mice to avoid changes, to avoid variability. These are the experiments I performed. So the first goal of my first experiment was to compare 4-EBP2 deamidation in the 2 parts of the neurons in the brain, the axons and the cell body. So I chose the retina and the optic nerve. Why? Because they are large extensions of the central nervous system, the diencephalon. They are easier to isolate than the brain, which is a mush of different brain cells. Their physical properties are identical to the rest of the neurons in the brain, so you can extrapolate And I isolated these axons in the optic nerve and the cell bodies in the retina. The cables of the brain cells are in the optic nerve. The cell bodies are in your eyes. This was the first time ever such an experiment was ever conducted. This was back in 2024. The second goal of my first experiment was to compare 4-ABP2 deamidation in the whole brain with the individually isolated axons in the optic nerve and the cell bodies, which led to my results. Deamidation is significantly higher in the optic nerve compared to its retinal ganglia. There is no significant difference between the deamidation of the optic nerve and the whole brain. There is a significant difference in deamidation between the whole brain and the retinal ganglia. And the conclusion was that deamidation is coming from the axons, at least in the central nervous system. I performed these same experiments in the peripheral nervous system to see if this was common throughout the entire body. So I looked at the sciatic nerve and the dorsal root ganglia, the ganglions in your spine. And same results. Deamidation was specifically coming from the axons. So, a comparison was also made between the sciatic nerve and the whole brain, and deamidation was much lower in the sciatic nerve than in the whole brain itself. So I compared the peripheral nervous system with the central nervous system. So it's not just the axons. There was something else going on here. So this previous result showed that not only is deamidation mainly in the axons, but it happens mainly in axons that have a kind of layer which protects them from the outside world. So this led to my theory. So there's three conclusions here. Deamidation is happening from the axons, conclusions 1 and 2, and deamidation is mainly happening in isolated axons. So this is a quick chemical flow sheet of deamidation. It's a molecule of asparagine which converts to an aspartate. And this principle extends to all asparagines in the body. 4-ABP2 deamidation occurs in asparagines 99 and 102 in this protein. The key to understand here is that, according to Arthur Robinson, the pioneer of deamidation, he found that deamidation is a clock. Deamidation has a lifespan. It takes a certain amount of time, always, for more than 50% of asparagines to become aspartates. And specifically, in 4-ABP2, it happens only and only 18 days after birth. So it's a clock. Based on these facts, I concluded that 4-ABP2 must have a protein half-life greater than 18 days for deamidation to occur. Let me explain. Proteins live a certain amount of time as well. You need 18 days after birth for deamidation to occur. If the protein lives 4 days, it's not going to reach the 18-day threshold necessary for deamidation to happen. If the protein lives 20 days, then it will reach that threshold. So, this was my first conclusion. The protein is living longer in the neurons. And my research led me to find that proteins in general, a related protein called sodium dismutase, had a significantly longer half-life, only in the axons. Proteins live much longer in the axons than in other parts of the body. Therefore, my conclusion was, 4-ABP2 lives longer in brain cells, which is why deamidation is unique to brain cells, and 4-ABP2 lives longer in brain cells because of the axons. So, we have Dr. Chehan over here. This was the link with the axons. Now I will establish the link with his amazing discovery he did in 1978 for the ubiquitin proteasome system. So, you have this system called the ubiquitin proteasome system, based on the 26S proteasome. This thing kills proteins. It degrades them. Axons are found to be proteasome-poor. Proteasome has been found to be higher in neurons without myelin. My experimental conclusion 3, if you remember, confirmed that deamidated 4-ABP2 is more enriched in the myelinated axons, where there's less proteasome of the whole brain, and significantly less enriched in the unmyelinated axons, where there's more proteasome in the sciatic nerve. Therefore, I concluded that the proteasome-poor environment in the axons is what makes 4-EBP2's half-life longer and deamidation thus possible. I also concluded that 4-EBP2 deamidation is maintained at different rates. And if you look here, this is a very interesting paper published in 2023. They did a fluorescence experiment. They isolated a brain cell, a neuron, and they looked at how the proteasome is spread throughout the axon and the cell body. And they found that in purple you have the proteasome. The proteasome is mainly located in the cell body. In green you have the rest of the proteins. There's very, very little proteasome in the axons. So there's very little degradation happening in the axons compared to the rest of the body. And this sealed the deal. A paper published in 2019. And unfortunately, these papers never made the connection. I made the connection in 2023, that if you inhibit the proteasome, this leads to an increase in the 4-EBP2 deamidation. The paper just never saw the link that the axons being proteasome poor led to 4-EBP2 deamidation, which was the big discovery here. You also had a confirmation that the only other part where they found 4-EBP2 deamidation was the muscles, where there's a huge enrichment of axons, axon terminals. Whereas in the heart, where there's no axon terminals, there was no 4-EBP2 deamidation. So this was my principle. Due to their proteasome-poor environment, axons increase the protein half-life, which becomes more significant than the deamidation half-life of asparagines, creating neuron-specific deamidation, which is a biological clock. So my contribution fundamentally changes the understanding of our protein production in the field. Axons are a control mechanism. They are deprived of proteasome, and this leads to an increase in 4-EBP2 deamidation. 4-EBP2 deamidation allows more proteins to be produced in an area that is practically devoid of devoid of DNA compared to the cell body. So 4-EBP2 deamidation in the axons is essentially a compensatory mechanism for a region which lacks proteins. For this region to survive, this region which sends information from one brain cell to the other is entirely dependent on 4-EBP2 deamidation, which was the conclusion of my paper. And this allowed me to establish the link to disease in my unified theory. So these are all papers cited in my review. Robinson et al. Described that deamidation leads to the programmed dysfunction of proteins. Pironosti et al. Discovered that deamidation occurs in 4-EBP2, which functions as a translational inhibitor. He also reported that 4-EBP2 deamidation reduces the speed and efficiency of this protein production. Tiwari et al. He discovered that 4-EBP2 deamidation decreases the speed of kinetics between one neuron to the other. Tiwari et al. Demonstrated that translational control dysregulation from 4-EBP proteins like SIFIB2 causes the early development of neurodegenerative diseases such as Alzheimer's. So if 4-EBPs don't work, you have a direct link to Alzheimer's. I discovered that 4-EBP2 deamidation is caused by the proteasome-poor environment in neuronal projections consisting mainly of axons. Cheng et al. And Dekoski et al. Reported that neurodegeneration occurs first in the axons and synapses in Alzheimer's, Parkinson's, and other neurodegenerative diseases. So the axons always degrade first. They're the main problem. Shiluri et al. And Hoshino et al. Reported that a decrease in proteasome activity in aging patients has been linked to the process of neurodegeneration due to pathological accumulation of proteins that are no longer degraded. Kululi et al. Found that deamidated 4-EBP2 overexpression is caused by proteasome inhibition and has been shown to decrease the expression of many genes implicated in cerebral cortex development and mitochondrial function, and that deamidated 4-EBP2 also decreases the activity of NF-kappa B in neurons. Remember this. This is very important. NF-kappa B is a survival protein. So the more 4-EBP2 you get, the less likely statistically your brain cell is going to survive. So it's necessary, but too much of it is dangerous. We'll get to that later. Sorrentino et al. Asserted that mitochondrial dysfunction has been found to contribute to neuron cell death in Alzheimer's and Parkinson's. Okay? And we saw a relation between this and 4-EBP2. Guo et al. Explained that mitochondrial dysfunction causes oxidative stress. Fang et al. Showed that axons are most vulnerable to oxidative stress. Sorrentino et al. And Fang et al. Stated that the reduction of dysfunctional mitochondria via mitophagy in neurons reduces pathological amyloid beta aggregation, inhibits pathological tau hyperphosphorylation, and reverses memory impairment caused by tau and amyloid plaques. So, you get mitochondrial dysfunction. This leads to amyloid beta, tau leads to Alzheimer's. You remove these dysfunctional mitochondria, the cell is healthier. Tain et al. Described that 4-EBPs inhibit PINK1 and Parkin protein mutants responsible for mitophagy disruption and dopaminergic neuron degradation in Alzheimer's. Kaltschmidt et al. Postulated that NF-kappa B protects neurons from necroptosis. Mefford et al. Reported that NF-kappa B activation depends on synaptic transmission and that NF-kappa B inactivation leads to learning and memory impairment. Tiwari et al. And Tong et al. And Li et al. Demonstrated that amyloid precursor protein and tau, as well as alpha-synuclein, all undergo cap-dependent protein translation involving EIF4E that is inhibited by 4-EBPs. Cruz et al. Reported that the excessive protein synthesis after 4-EBP inhibition results in neurodegeneration in diseases such as Huntington's disease. This is a lot of information which I synthesized in one figure, which is my unified theory. You have a direct link here. When you have excessive protein production, which is what the 16 papers explain, you have excessive production of dysfunctional mitochondria, of alpha-synuclein, of tau, of amyloid precursor protein. This was proven in 16 papers. And why is there this accumulation? There is a lack of proteasome production as you get older, and this principle extends to all mankind. So, as you get older, proteasome activity goes down. Usually, this would be compensated by 4-EBP2. You'd have less proteasome, you'd have more APP2 to reduce protein production. But that is not possible in the brain specifically, because 4-EBP2 gets deamidated. The more 4-EBP2 you have, the more deamidation you're going to have. It always happens after 18 days. And 4-EBP2 specifically in the brain, as you get older, cannot compensate for the accumulation of these pathological proteins. More than that, 4-EBP2 slows down transmission of information to a certain degrees between neurons. That's a good thing when you're healthy. But as you get older, this inhibits the survival protein NF-kappa B to a certain degree. Too much accumulation of proteins leads to dysfunctional mitochondria, which inhibits the recycling of dysfunctional mitochondria, causes oxidative stress, and leads to Alzheimer's and Parkinson's. So you have a wave of different biological pathways that are impacted by excessive 4-EBP2 as you get older. My unified theory establishes that the key here is to target 4-EBP2 deamidation. If you are able to slow down 4-EBP2 deamidation as the patient gets older, you will have more 4-EBP2 to compensate for the increased protein production caused by a lack of proteasome, of UPS. And this will stop the accumulation of proteins such as tau, and this will lead to less neurofibrillary tangles, amyloid beta plaque, which will lead to an effective treatment and in some cases even reversal of Alzheimer's. There was a famous paper in one of the 16 that I presented that showed a direct link between hyperactive EIF4E and hyperphosphorylated tau. Hyperphosphorylated tau is one of the main pathologies of Alzheimer's. Hyperactive EIF4E can be directly neutralized by 4-EBP2. So this is my talk. Thank you so much. and I was on time.
Thank you, David. So this is again, this is a unified theory in terms that you have a switch that can control numerous neurodegenerative diseases like Alzheimer's, Parkinson, and others by controlling this switch, by making it work in specific ways. So which is sustainable because reduces a lot of the resources, increases the effectiveness, and so on.